In this tutorial, we'll have a quick look at phototropism. Phototropism is the movement of plants, the stems of plants, towards a light source. So it's a positive tropic response. It is a movement towards the stimulus. It fits the stimulus response model that we've explored in class. Here's a couple of photos you can see of this phenomenon. So we have plants bending towards the sunlight through the window. Again, we have small shoots, coleoptiles, moving towards a light source. So let's have a look at the mechanism for phototropism. So a stimulus is a light source. The receptors that detect the stimulus are proteins embedded in the membranes of the cells in the tip of the plant. The cells then produce a hormone messenger, auxin, in particular the auxin indole acetic acid, IAA, which moves down the side of the plant on the far side of the light source and causes the cells down this side to elongate, to grow longer, and this in turn causes the response of the bending of the plant towards the light source. So the stimulus is the light. The receptors are protein receptors in the membranes of the cells in the tip of the plant. The effectors are the cells on the side away from the light, and the response is to bend towards the light source. So let's look at this mechanism in just a little bit more detail. What is it that actually detects the stimulus, and what exactly is the stimulus in phototropism? It's been discovered that the stimulus for phototropism is in fact the blue wavelength of light, and we'll have a look at this in a graph in just a moment. So the blue wavelength of light is the stimulus and is detected by special proteins embedded in the cell membranes called flavoproteins, and it's particularly the flavoprotein phototropin. And it's this particular protein that brings about the auxin transport and therefore the elongated growth of the cells on the far side of the plant. So let's have a look at first of all the graph and then how this might work in a little bit more detail. In this graph we can see the degree of phototropic response at different wavelengths of light. So we can see that the y-axis is measuring the amount of phototropic curvature, and the x-axis is showing the different wavelengths of light from the violet end of the spectrum down to the red. And we can see quite easily that most phototropism, most phototropic curvature here occurs in the range of the violet through to the blue, somewhere between about maybe 420, 430 up to about 500 nanometers. There is some curvature in the violet end down here, but the majority of it is in the blue area. We can see that here. We can see the plants bending in blue, but in the green, orange and the red wavelengths there is no bending of light. So the receptors for phototropism are blue light receptors, so they're capable of detecting the blue wavelength of light only. Let's look at the mechanism by which this might work. If we were to look at this at the cellular level, we're looking at the cells that are in the tip of the plant, and it's these cells that have the phototropin proteins embedded in their membranes. And it's phototropin that is capable of detecting the blue wavelength of light. So presumably that as the blue wavelength of light hits the phototropin, this sets off a signal cascade, which in turn will bring about a series of reactions in the cell that will result in the production of indole acetic acid, or auxin hormone. So this will be synthesized, and the auxin will be secreted by these cells and will travel down to the cells on the far side of the plant. So if we were to see this kind of effect, the cells on the far side of the plant will presumably have receptors in their membrane for auxin, so they will have particular protein receptors that the auxin molecules are capable of locking into. This in turn will set off another signal cascade, further signal transduction, which will ultimately lead to the response of these cells then growing longer, which in turn will cause that side of the plant to extend out and hence push the stem over towards the light source. Let's have a look at this final step in the next section. So here we will see our light source, our original plant. The cells down this side of the plant will grow longer in response to IAA or auxin. The cells on this side of the plant will remain the same size and will not elongate. So this side of the plant will require effectively more space than this side of the plant, and this will push the stem over towards the light. One presumes that in terms of evolutionary survival, that this is about increasing the light intensity for photosynthesis, hence the plant can make more carbohydrate for growth. So to sum up, the stimulus will be the blue wavelength of light. The receptor will be phototropin proteins embedded in the membrane of the cells in the tip of the plant. The messenger molecule will be indole acetic acid, which is a kind of auxin hormone. The effectors will be the cells on the far side, away from the light of the stem, and the response will be the bending of the stem towards the light source.